in cell structure and functions. The next part is biomolecules. So let's start with the introduction to the biomolecules. So what is a biomolecule, or what are biomolecules? So before that, we know that cells are made up of thousands of chemicals. So those chemicals which are present in a cell, which forms the major constituent part of a cell, are called as a biomolecules. So here these biomolecules are the one which are made up of carbon, hydrogen, oxygen, nitrogen. Apart from these elements, various other type of elements are also be the constituent part of the cellular components. So that forms the biomolecules. So in this first let's see the what is cellular pool in a cell. Cellular pool. So the sum total of chemical elements present in a cell is called as the cellular pool. That means whatever the chemicals which are present as a constituent part of a cell, that total number of elements or the substances or the molecules or ions, so that entire structure is called as a cellular pool. So this cellular pool will exist in two states or two phases. One is called as aqueous state and other is called as non-aqueous state, or aqueous phase and the non-aqueous phase. Aqueous phase means the chemicals which are present, or the ions, the elements which are present, these are dispersed or some may be dissolved in the water content or the liquid content. Instead of saying water, if we say liquid content of the cell, that forms the aqueous phase of cellular pool. And all these are existing in all these aqueous elements will be existing in colloidal state or colloidal form. Some may be dispersed or little bit we can see them are microscopic structures which are not dissolved, and some of them are dissolved. So such type of non-dissolved or dispersed one are called as a colloidal in their nature, and such a state is called as colloidal state. Then what is non-aqueous state or non-aqueous phase? Non-aqueous phase includes the chromatin, the elements present in the chromatin part, the one which are present in the cell membrane, and the chemicals which are present in the cell wall, which is not dispersed. Such a quantity or such a quantum of the cellular pool is called as a non-aqueous phase of cellular pool. Likewise, so this is about the elements which are present, but which type of elements are present, how many elements are present. So that are to be analyzed by various techniques. So if we take a living system or any living organism or cell of any living organism, like it a plant, an animal or a microorganism. So if we want to know what how many elements are present or what are the elements that are present in a cell, so that have to be done by elemental analysis. In this elemental analysis, we are going to know that what are the elements that are present in a cell. So that is what we say knowing about various types of elements or knowing about the elements which are present in a cell. So in this process we know about the elements, but what about what are the elements which are present in a living system or in a living cell, and what are the elements which are present in a non-living system? Let's take the example as earth crust. Consider that the living system contains carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, potassium, etc. The same way, if you analyze the non-living system like earth crust, that also contain the same type of elements, but the quantity of the element which are present may be differing, but basically the elements which are present will be nearly similar in both the living system and also in the non-living system. And where does the difference will come is the quantity of the elements which are present. Basically, the abundance of carbon and hydrogen is more in the living system or the living cell when compared to a non-living system or a earth crust. So likewise, we know about the elements which are present in the living and a non-living system by a process called as elemental analysis. Then chemical analysis. After knowing about what are the elements which are present and which type of elements that are present in a cell or present in a living system or present in a non-living system or a earth crust. So that is analyzed by a technique called as a chemical analysis. In this process, if you take a living part of any plant or a animal, like a liver part of a animal, and that have to be crushed, or we can say it have to be grinded by using trichloroacetic acid. Trichloroacetic acid that have to be grinded, and we get a thick slurry. So that slurry have to be stained through cheese paper or some cotton filtrate like thing, so that we get two parts. One is called as filtrate and other is called as retentant. Filtrate is called as acid soluble fraction, whereas retentant is called as acid insoluble fraction. Likewise, we get two parts: filtrate or acid soluble fraction, retentant or acid insoluble fraction. The scientists had analyzed that presence of various types of elements or various types of organic compounds present in the filtrate. They find that thousands of organic compounds are present in acid soluble fraction. Of course, it is not only that organic compounds are present in the filtrate part, but many organic compounds are present in the acid insoluble fraction also. Simply to say, the one which have lower molecular weight are present in the acid soluble fraction, whereas the organic compounds which contain higher molecular weight, or simply to say more than 1000 Daltons, all such organic compounds are present in the acid insoluble fraction. By this we know about what type of chemicals that are present in a cell or which are present in a living body or present in a non-living body also. That process is called as, or that technique is called as chemical analysis. Next is separation technique. When we do the chemical analysis, we know that what type of chemicals are present, but so it is not like that the chemicals or the organic compounds are separately present in a cellular pool. They are mixed with each other. So how can we know what is the actual nature of a organic compound or a element or any substance? Means by separating them from each other. That have to be done by a technique called as separation technique. So after separation technique, so we have to go for ash analysis, so that we can know what is the actual element, what is the actual compound, what is the actual molecule, ion that is present in that particular substance or the particular part of a plant body or a animal body. So that is done by ash analysis. And before that, what is a biomolecule? As already we said that all the chemicals which are present in a body or which are present in a living body or a living cell or a non-living body or any earth crust. So all that is called as a biomolecule. Otherwise, we can say that a chemical or a substance which is made up of basically by carbon, hydrogen and oxygen. So those are considered to be the biomolecules, but it is not compulsory that always they should contain only these three elements. Sometimes they may also contain additional elements also, sometimes they may not contain some of these elements also. Like it is not a particular type of element that have to be present in a biomolecule. Then how we have to go for ash analysis? If we take any part of a plant body, we have to dry it. When we dry the plant part, whatever the liquid content that is present in the plant cells will dries off. So then we get a dry part. So that is what we call it as a dry weight of a plant. After evaporation of all the water or the liquid content that is present in the plant cells, whatever the dry material which is left is called giving the weight, that is called as a dry weight. And here we have to know something about fresh weight also. So here fresh weight means weight of a plant body along with the aqueous materials present in them, that is called as a fresh weight. That means not only the weight of cell organelles, not only the along with the cell wall plasma membrane, and also various liquid contents which are present in the cell, all that includes the fresh weight of the plant. So let's come to the dry weight here. So after that we have to burn the dry part of the plant body. When we burn the dry part of the plant body, we know that some fumes will come or some sort of soot will be coming out. So here in this process, some amount of water vapor will be lost. Little amount of water which is present. Already dry weight means it contains very majority of the water is lost, but very few quantity of water will be left out. That is also been lost when it is burnt, and also the carbon dioxide which is released outside. And after this, whatever we get is the ash. When we analyze the content, substances which are present in the ash, we come to know about what is the type of element or the substance which is present. So it may be an individual element or a compound. So individual element means it may be magnesium, calcium, phosphorus, sulfur, like individual elements. If it's a compound, compound means like sulfate, nitrate, such type of phosphate, such type of structures are called as a compounds. So by doing this ash analysis, we know about various types of elements which are present, and also the substances which are present in the plant body or any living system. So likewise we know about various types of elements, what type of element is present and in which combination they are present, and how to separate them. All these techniques will be the part of the introduction of the biomolecules.